Okay, exercise 16 is a big lab. There's lots to inoculate, so I've split it up into two videos here. The first video, I'm just going to talk about the organisms a little bit, as well as one of the medias you're going to inoculate, and then the second video will be the other 5 tubes that you're going to inoculate. Okay, so we're in the home stretch. You're going to be thinking about gram-negative identification here in this lab. Okay, so if you look at page 70 on your data table there of your lab book, you'll see the 10 organisms that you are going to learn to distinguish between in this lab and the next one, exercise 70. Okay, so what do all these organisms have in common? They are all gram-negative, rod-shaped organisms. Okay, now a lot of times students kind of get into the habit of thinking all gram-negatives are rod-shaped. That's not the case. There are gram-negative cocci. They're not as common, and they're much more difficult to grow in the lab, and a lot of them are some nasty pathogens, like gonorrhea, for example. So we're not going to be growing any of those in the lab, but all of these that we're working with just happen to be rod-shaped. What else can we say that these 10 organisms have in common? Well, they're all mesophiles, means they grow best at room to body temperature. They are all non-spore formers. Remember, that's not very common in the microbial world. Mostly Bacillus and Clostridium that are the spore formers. So even though these are rod-shaped, they're not spore formers. Most of these 10 organisms are in the same family, and that's Enterobacteriaceae. So most in the family Enterobacteriaceae. Kind of a mouthful. I'm underlining it because it's grammatically correct to either underline or italicize family names, just like genus and species. So remember your levels of taxa. The lowest level is species, next up is genus, next up above that is family. Okay, so this is a group of organisms. You may not have heard this term Enterobacteriaceae before, but probably at some point you have heard the term enteric. Okay, we usually abbreviate Enterobacteriaceae and just call them enterics. Okay, and that is from the Greek root enteron, which means intestinal. Okay, so you may have heard of enterotoxins or enteroviruses. Okay, that prefix gives you a hint that this is the business of this molecular organism happens in the intestine. Okay, so 8 of these 10 organisms listed on page 70 are in this family, and that means they are all found as normal microbiota in the intestine, especially large intestine, or they are found there when we eat some contaminated food or drink some contaminated water. Like salmonella, for example, is an enteric organism. Not part of your normal microbiome, but definitely we ingest some bad chicken or whatever that's not been cooked, and definitely that's where the organism attaches and does its work. So I'm not going to go through this list one by one like I did for our gram-positives. You'll notice just a mix of genera. We have a couple of Proteus organisms and a couple of Klebsiella, and just kind of a mix of a lot of different things. I would say what they have in common is I would call most of them opportunists. Or in other words, they're opportunistic pathogens. And what that means is that they typically are not going to cause disease in humans unless certain circumstances arise. Okay, so for example, Enterobacteriaceae. That's an organism that's normally found in our large intestine. It's not going to cause us any harm unless, say, the bowel is perforated and those organisms, get out into our body cavity or get in the bloodstream. Okay, then yeah, they're going to cause some problems, some disease. Okay, so typically, unless the patient is immune compromised or there's some kind of wound infection, again, the organism gets somewhere in the body that it doesn't normally belong, then it's not going to cause a problem. Although most of these organisms are able to cause urinary tract infections in a healthy individual. Okay, actually, E. coli is, we think, the number one cause of UTIs, but there are probably at least a couple hundred organisms that cause urinary tract infections. Okay, think about it. If all these organisms are normal microbiota in the large intestine, that means they are going to make up part of our feces, and especially if somebody wipes back to front instead of front to back, that's going to give just perfect opportunity for that urinary tract infection. Okay, so again, much less commonly septicemia. That's when an organism, bacteria, invades the bloodstream. Okay, that's a very serious situation. It can happen, maybe after a surgery or very, systemic infections. Mortality is about 50% when that happens. A couple of these organisms can cause pneumonia, although not commonly, like Klebsiella pneumoniae can cause pneumonia. Pseudomonas is known to cause pneumonia. Pseudomonas is a little bit better known for causing hospital-acquired infections. So again, maybe you have a burn victim where their skin has been burned off. That's our first line of defense that's gone, that's given the organism a chance to cause infection where it normally wouldn't in a healthy individual. Okay, next I want to talk about our selective media that we have that only gram-negatives will grow on. Okay, and that's called McConkey Agar. So remember, we had a selective media that only gram-positives grow on. That was the CNA blood agar plate. So if you had a mix of organisms, say some gram-positives and gram-negatives, if you streak them on McConkey Agar, only the gram-negative will grow. And we don't actually test that in this lab. We assign you a gram-negative organism and you streak it out on McConkey and we see what it looks like. But if you look at the recipe for McConkey Agar, what makes it selective is the crystal violet and the bile salts. Okay, so the crystal violet, that's the same dye that we use when we're gram staining. That maybe gives this a little kind of pinkish hue. I'm not sure, but this is McConkey Agar over here. It's not a great picture. And then bile salts, again, there's something about that combination of crystal violet and bile salts that most gram-positives can't tolerate. Okay, so those are what we would say the selective agents. Okay, only gram-negatives grow. Now the interesting thing about this media is that it is also a differential medium, because there's lactose in there as well as the pH indicator, neutral red. Okay, so with those two ingredients, again, these are differential agents. Okay, a sugar like lactose, that's not going to keep an organism from growing. But if an organism can use lactose, can ferment lactose, then what do we get? when an organism ferments a sugar, we always get acidic end products. Okay, so organisms that use lactose, the colonies and the media, let's see, the colonies and to some extent the media around it is going to turn bright pink. Okay, I'll show you an example of that in a minute. Okay, so with this pH indicator, you don't have to know these numbers, I don't have this memorized, but between about 6.8 and 8.4, we'll call that neutral, whatever this color over here is, it's kind of pinkish. Let's see, above 8.4, it gets to be kind of, this is a hard color to describe, kind of tannish, maybe colorless, not totally clear, but a little tannish, kind of yellow, and then below 6.8 in acidic pH, it gets to be usually kind of a hot pink. Okay, sometimes that looks a little bit more red, depending on what else is in the media, but okay, so there are other things in the media here that aren't quite as interesting. You got a little tiny bit of salt, okay, not enough that makes this selective, just a little pinch for osmotic balance. I didn't write in the amounts here. You've got, of course, a little bit of agar that makes it a solid media. Okay, so in this lab, you're just going to streak out normal 4-quadrant streak plate technique with your assigned organism. Really important, remember, to try to get as many isolated colonies as we can, just so we can check real carefully and make sure there's only one organism growing on there. Okay, so again, that will be really important when you're doing your unknown. Later on, you need to make sure that you're only doing tests on one organism. You would never want to do any of these tests that I'm going to talk about next in the next video on a mix of bacteria, because you would get conflicting results. Okay. All right. So, here is an example of a lactose negative organism. So with that pH indicator neutral red, the colonies are, I don't know, what color would you describe that? I don't know, they're kind of brownish, yellowish, almost tannish. And then here's an example of a lac positive organism. Okay, so you know both these are gram-negatives because it wouldn't grow on MacConkey if it wasn't gram-negative. Now, some of the organisms are really not this obvious. Okay, there's a few organisms where the colonies are actually a little bit pink, but because the background is not, we don't consider it lac positive. So I want you to be really careful when you're looking over the results of the MacConkey test and make sure you get to see all the organisms, just so you get a little practice discerning the lactose positive from the lactose negative. Okay, so watch the next video. I'll talk about the IMBIC test as well as the urea tests. These tests are going to help us identify a gram-negative rod.